Can aging be stopped? The answer from physics is split in two, because aging itself turns out to be two completely different things happening inside your body at the same time, and almost nobody separates them. One is thermodynamic. Your body burns 70 watts of continuous power, the output of an incandescent light bulb, every hour of every day, devoted entirely to keeping you intact. 10,000 DNA lesions in every cell, every day, detected and repaired. Millions of sodium ions pumped across every neural membrane every second to maintain the voltage difference that allows your brain to process this sentence. Damaged proteins identified, dismantled, recycled, rebuilt. You are a machine fighting the second law of thermodynamics with every breath you take, and after 60 or 70 years, the second law is winning. That part of aging cannot be stopped. But the other part is not physics at all. It is biological programming, deliberate instructions your genome follows to shut down repair systems, shorten cellular clocks, and slow energy production on a schedule that was written for a species that rarely survived past 40. That part is software. There is an animal alive in the North Atlantic right now, born before Newton, whose cells carry a different version of that software. And in 2012, researchers at Harvard ran an experiment on mice that were old and frail and cognitively declining. And those mice got younger. To understand why that matters for the body you are living in right now, you need to understand what it reversed. Entropy is the word physics uses for disorder. And the second law of thermodynamics states that disorder increases over time in any physical system. Your body fights this by spending energy on maintenance, and the scale of that maintenance is difficult to overstate. The 10,000 DNA lesions per cell per day are not random. They include single-strand breaks from oxidative stress, double-strand breaks from replication errors, and base modifications from environmental chemicals, each requiring a different repair pathway with its own specialized enzymes. Your protein quality control runs 3 parallel systems: chaperones that prevent misfolding before it starts, proteasomes that shred damaged proteins into recyclable amino acids, and autophagy, a process where the cell literally digests its own damaged components inside membrane-bound compartments. All of this runs continuously in every cell, without rest, for your entire life. 1/5 of everything you eat goes to nothing but maintaining the voltage differences across your neural membranes, the electrochemical gradients that let your neurons fire. That is the metabolic price of being alive and conscious. You have been paying that price every second of every day for your entire life. And the machinery doing the work is wearing down. But the maintenance machinery is itself made of molecules, and molecules accumulate damage. Over decades, the repair enzymes develop their own copying errors. The quality control systems lose precision. The cost of maintaining order rises, more energy spent achieving less repair. This is the thermodynamic floor of aging. It is the second law expressed in biology, and nothing will ever push below it. Stars age. Mountains erode. The universe itself is aging toward a final cold equilibrium so distant it makes all of human history look like an opening breath. Your body is no exception. If aging were only thermodynamics, the story would end here. You would wear out gradually, uniformly, and there would be nothing to discuss except the physics of inevitable decline. But in 1961, a biologist saw something that did not fit, and what he found changes everything that follows. Leonard Hayflick was culturing human cells at the Wistar Institute in Philadelphia. The standard assumption in biology at the time was that normal human cells, given nutrients, would divide indefinitely, that cellular immortality was the default. Hayflick watched his cells divide, and they did not behave that way. They divided roughly 40 to 60 times and then stopped. Every culture, every cell line, the same limit. He repeated the experiments. He controlled for contamination. He varied the nutrients. The limit held. His colleagues told him his technique was flawed, that healthy cells should be immortal in culture. Hayflick disagreed. He believed the limit was real, built into the cells themselves, a feature, not a failure. He published in 1961 faced professional skepticism that lasted years and was eventually proven entirely right. What Hayflick had discovered was the first evidence that aging is not just thermodynamic wear. The cells were not breaking down from random damage. They were counting their divisions and stopping on a schedule, as if something inside each cell kept a tally and, upon reaching a predetermined number, pulled the brake. Something was acting as a clock, and the clock was not optional. That changes the question entirely. Because if aging is partly a program, a set of instructions the cell follows, then the question becomes whether the instructions can be changed. What Hayflick had found was the telomere, a repeating sequence of DNA bases that caps the end of every chromosome the way a plastic tip caps a shoelace. Telomeres contain no genes. They are protective buffers. Every time a cell divides, its telomeres shorten slightly, because the molecular machinery that copies DNA cannot replicate the very last nucleotides of a linear strand. This is a geometric limitation, not a flaw, a physical consequence of how DNA polymerase works. After 40 to 60 divisions, the telomeres shorten to a critical length, and the cell either stops dividing, entering a state called senescence, or triggers its own destruction. Hayflick's limit, explained at the molecular level. That counter is in every cell of your body. It has been running since before you were born. Your immune cells have divided thousands of times across your lifetime to fight every infection you have ever had, every cold, every flu, every wound that reddened and then healed. Your skin has replaced itself roughly every 2 to 3 weeks for 60 or 70 years. Over 1,000 complete turnovers of the largest organ in your body. Your gut lining has renewed itself every 3 to 5 days, building a new intestinal surface from scratch more than 5,000 times since you were born. Every one of those divisions shortened the telomeres in those cells by a measurable amount. The cells in your body at 65 carry quantifiably shorter telomeres than the cells you had at 30, and the shortening varies by tissue. Your immune cells, which have divided the most, show the most shortening. Your brain neurons, which rarely divide, retain longer telomeres. The counter Hayflick discovered is in your body. It has been running your entire life. And it has run different distances in different tissues depending on how hard each tissue has worked. An enzyme called Telomeres can rebuild telomere length, can add the repeating sequences back to the chromosome ends, and effectively reset the counter. It operates in your stem cells and your reproductive cells, which is why those lineages can divide far more times than ordinary tissue. In most of your body's other cells, telomerase is deliberately suppressed. The reason is a trade-off that evolution made. Unrestricted telomerase activity is precisely the mechanism cancer cells use to achieve immortality. Cancer reactivates telomerase and divides without limit. Your body suppresses telomerase in most cells to keep cancer in check, accepting a finite cellular lifespan as the price of tumor suppression. Elizabeth Blackburn and Carol Greider shared the 2009 Nobel Prize for discovering telomerase and understanding this trade-off. Greider was Blackburn's graduate student at the time. They found the enzyme in tetrahymena, pond scum, the molecular key to cellular aging discovered in single-celled organisms from a pond. Everything up to this point tells you that aging has two layers, a thermodynamic fall that cannot be changed, and a programmatic clock that counts down towards cellular shutdown. If you stopped here, the honest conclusion would be that the clock is a one-way mechanism. It ticks, the telomeres shorten, the cells stop, and nothing reverses it. That is what biology believed for decades after Hayflick. Then Harvard ran the experiment. In 2012, researchers at Harvard Medical School reactivated telomerase in mice that had been engineered to age prematurely. These mice were the equivalent of elderly humans: shrunken brains, organ degeneration, cognitive decline, thinning fur, reduced fertility, poor wound healing. They were old, measurably, visibly, functionally old. After telomerase was reactivated in their cells, the aging reversed. The spleen, the liver, the intestines returned to normal size and function. Brain volume increased. Cognitive performance improved on maze tests. Fertility returned in animals that had been infertile. The mice did not simply stop aging. They got younger. The clock had been wound back. Let that settle. Hayflick discovered the counter in 1961. For 50 years, biology assumed it was one way. Cells divide, telomeres shorten, the counter runs down, nothing reverses it. The Harvard experiment shattered that assumption. Hayflick's clock, the same telomere shortening mechanism he discovered, was reversed by reactivating a single enzyme. The mice did not receive young organs or stem cell transplants. Their own cells, given back the enzyme that had been suppressed, rebuilt their telomeres, resumed division, and restored tissue that had been deteriorating for months. The hardware for reversal was there all along. It had been switched off by the same genome that built it. This has not been replicated in humans. The cancer risk of broadly activating telomerase in a human body is the central unsolved problem. The same enzyme that reverses aging is the enzyme cancer uses to become immortal. But the experiment proved something no theory could. The biological clock is not a one-way ratchet. Under the right conditions, it runs backward. The question the entire field is now organized around is how far backward it can go and in how many systems. Now, the telomere experiment showed that Hayflick's clock can be wound back. That is radical enough. But the clock is only one system. The question Hayflick's discovery opened, can the instructions be changed, turns out to go much deeper than telomeres. Because there is a layer of instructions sitting on top of your entire genome that does not just count divisions. It controls which genes are active, which are silenced, which repair systems run, and which shut down. And it changes with age in a pattern so consistent that a scientist at UCLA built a clock out of it. You can feel this layer working. Each of your heart muscle cells contains up to 5,000 molecular turbines, called ATP synthase, spinning motors, nanometers across, that rotate at 9,000 revolutions per minute to produce the energy your cells run on. If you are 65, those turbines have powered roughly 2.5 billion heartbeats without a single pause, one of the most sustained physical performances in the known universe. I find that remarkable. After roughly age 40, their output declines. You know the feeling, the specific afternoon dimming that is different from tiredness, more like the light in a room going slightly flat. That decline is not just mechanical wear on the turbines. It is partly regulated by changes in the instructions sitting on top of your DNA. Epigenetic modifications that suppress the genes controlling mitochondrial production. The hardware still works. The instructions are telling it to slow down. In 2013, a biostatistician named Steve Horvath at UCLA published a discovery that gave aging research its most precise measuring tool. Horvath developed a method of estimating biological age from the pattern of methyl groups, chemical tags, sitting on top of the DNA in any tissue sample. The epigenetic clock. It is accurate to within a few years, and it does not always match the calendar. Two people born in the same year can have Horvath clock ages that differ by a decade or more. One biologically 55, the other biologically 65, despite identical birth certificates. That gap is not random. It maps to real functional differences. Tissue repair speed, immune response strength, cardiovascular resilience, cognitive maintenance. Your biological age is the age your cells are actually operating at, and it reflects the cumulative effect of everything your body has been through. Your body carries a biological record of your entire life in this layer. Every decade of physical activity wrote lower methylation at inflammatory genes, keeping them quieter, keeping tissue inflammation lower. Every year of adequate sleep wrote into circadian gene accessibility, maintaining the rhythms that coordinate repair across organs. Every period of chronic stress wrote higher methylation at stress buffering genes, making them harder to access, making recovery slower. The record is specific. It is readable with current technology, and it is accumulating in every cell of your body as you listen to this. Your body has been writing this record for your entire life. The question is whether any of it can be erased. In 2020, David Sinclair's laboratory at Harvard did something to that record that changes the picture entirely. They took aged mice with deteriorating vision, retinal ganglion cells loaded with decades, equivalent of accumulated epigenetic marks, and introduced 3 genes called the Yamanaka factors that partially reset the epigenetic state of those cells. They did not change the DNA sequence. They changed the marks sitting on top of it, rolled the epigenetic program back toward an earlier version. Sinclair's cells returned to a younger epigenetic state. The mice's vision improved, measurably, functionally. Aged eyes began to see again. The DNA had not changed. The instructions running on top of it had been rewritten, and the cells obeyed the rewritten instructions. Think about what that means for the cells in your own body. Some of the decline you experience may not be irreversible, thermodynamic damage. It may be epigenetic programming, instructions your cells are following that could, in principle, be revised. Your cells may be acting older than their DNA requires because accumulated epigenetic marks are telling them to slow down, reduce repair, operate in a degraded mode the underlying hardware does not demand. Telomeres showed Hayflick's clock can be reversed. Epigenetics showed the entire operating program can be rewritten. But both of those address the instructions going forward, what the cell does next. There is one more mechanism, and it addresses something different, the damage that the old instructions have already caused, the wreckage that has been accumulating in your tissue for years, and it connects to something you feel in your body every morning. When a cell reaches the end of its telomere capacity and stops dividing, it enters senescence. A senescent cell does not die. It persists in the tissue, occupying space, consuming resources, and doing something actively harmful, secreting a cocktail of inflammatory molecules called the senescence-associated secretory phenotype, a continuous broadcast of signals that damage surrounding healthy cells and accelerate their aging. Researchers call them zombie cells, not dead, not functional, actively toxic to their neighbors. In a young body, the immune system clears them efficiently. In an older body, clearance slows and they accumulate. In joints, in lungs, in kidneys, in skin. A background ache that many people over 65 carry as a constant. The joints that never fully settle, the mornings that take longer to start, the recovery that used to happen overnight and now stretches into days, the general sense that your body is running a degree off from where it used to has a measurable cellular component. Stop cells, still embedded in the tissue, still broadcasting inflammatory signals to the healthy cells around them. Your body is not only wearing out from thermodynamic decay, it is being actively degraded by its own accumulated zombie cells, sitting in your joint cartilage, your lung tissue, your kidneys, your skin, sending distress signals that make the surrounding tissue age faster. If you feel that low-grade inflammation, the ache that is not an injury, the stiffness that is not from exertion, part of what you are feeling is the cumulative broadcast of cells that stopped dividing years ago but never died. The science can now see them, count them, and is learning to remove them. A class of drugs called Senolytics, currently in human clinical trials, selectively eliminate senescent cells while leaving healthy tissue intact. In mice, senolytic treatment extended healthy lifespan, reduced physical decline, and improved function in joints, lungs, kidneys, and cardiovascular tissue. The zombie cells can be identified, targeted, and cleared. Here is what the science now shows. Each result answering the question Hayflick's discovery opened in 1961. He found the clock. The telomere experiment showed the clock can be wound back. The mice at Harvard got younger. Their organs regenerated, their brains grew. The epigenetic experiment showed the operating program can be rewritten. The blind mice at Sinclair's lab saw again their retinal cells behaving as if they were decades younger. The senolytic experiment showed the accumulated damage can be cleared. The inflammatory signals reduced, the tissue degradation slowed. 3 separate mechanisms of aging, each one programmatic rather than thermodynamic, each one at least partially reversible in laboratory animals. None of these is the second law of thermodynamics. All of them are biological programming. And biology, unlike thermodynamics, responds to intervention. Tom Kirkwood at Newcastle University explained why this programming exists. His disposable soma theory, published in 1977, describes a trade-off natural selection made on your behalf. In a world of limited resources, an organism must choose between investing energy in reproduction and investing energy in long-term bodily maintenance. For most of human evolutionary history, external threats killed individuals long before cellular aging mattered. There was no payoff for maintaining a body past 40. Natural selection optimized for fast reproduction and minimal long-term repair. Evolution designed your body to reproduce, not to last. The fact that you are still alive at 65, still thinking, still maintaining voltage gradients across 100 billion neurons is an engineering achievement that exceeds the design specification by decades. The shutdown instructions your genome carries were written for a shorter world. They are still running in a longer one. Nature has already demonstrated that the biological clock can run at radically different speeds. The Greenland shark, Somniosus microcephalus, lives for approximately 400 years. A specimen caught in 2016 was radiocarbon dated to roughly 392 years old, born around 1620, when Galileo was still publishing and Newton had not yet been born. 4 centuries in the same body, in the deep cold water of the North Atlantic, its metabolism running near freezing, its cell division rate slowed to a fraction of what warm-blooded vertebrates experience, its thermodynamic cost stretched across a span of time that exceeds the entire history of modern science. Another proof comes from the naked mole rat. It lives roughly 30 years, 10 times longer than other rodents of similar size, and shows a property called negligible senescence. Its mortality rate does not increase with age. A 25-year-old naked mole rat is no more likely to die than a 5-year-old one. Its protein quality control systems, the chaperone networks that prevent misfolding, the proteasomes that clear damaged molecules, maintain precision far longer than those of other mammals. And it almost never develops cancer, despite carrying cells that divide throughout its long life. The jellyfish Turritopsis dohrnii can revert from its adult form to its juvenile state under stress, resetting its developmental clock entirely. These are proofs of concept. The biological programming of aging is not universal, not fixed, and not the only solution life has found. Different organisms solve the same thermodynamic problem with different strategies. The human strategy was optimized for a world of predators, infection, and famine, a world that no longer exists. Somewhere in the North Atlantic, a Greenland shark is gliding through cold, dark water. 4 centuries old, born before Newton discovered gravity, carrying in every cell a biological solution to aging that evolution never gave the human genome. That shark is alive. The mice at Harvard got younger. The retinal cells at Sinclair's lab began to see again. The zombie cells in the mouse joints were cleared and the inflammation dropped. Here is where the line falls. The thermodynamic floor cannot be moved. Stars age. The universe ages. Nothing built from atoms escapes the second law. But the biological programming sitting above that floor, the telomere clocks counting down, the epigenetic marks telling your cells how old to act, the zombie cells broadcasting inflammatory signals, that layer is not physics. It is evolution's compromise, written for conditions that no longer apply, running in a body that has already exceeded its design window by decades. The question is whether science can renegotiate that compromise in a human body, whether the epigenetic marks that told a mouse retinal cell it was old can be reset in a human eye, whether the zombie cells in your joints and lungs can be cleared as effectively in your tissue as they were in a mouse, whether the telomeres that reversed Hayflick's clock in a Harvard laboratory can be activated in a human body without activating cancer. None of these questions has been answered for humans yet. All of them have been answered for mice. The distance between a mouse answer and a human answer is where the next decade of science lives. The atoms in your body are approximately 13 billion years old. They have survived supernovae. They have been part of stars, oceans, rock, and 1,000 other living things across spans of time that make a human lifetime invisible. This particular arrangement of them, this specific person, this specific body, has existed once and will not exist again. How long that arrangement holds is partly thermodynamics, which cannot be negotiated, and partly biology, which is being negotiated for the first time in the history of the species. The answer is not yet written. For the first time, that feels like a genuine opening.